Hello, everyone. Welcome to today's live broadcast, The Serious Science of Schmutz, Deep Sequencing the Built Environment. I am Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars, contributing to the advancements of science through content sharing capabilities. LabRoots is a powerful advocate in amplifying global networks and communities. For more information, please visit labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by tapping on the Q&A box, which can be found on the Q&A box on the lower left hand of the screen. We'll try to answer as many questions as we possibly can. You can enlarge the slide window by clicking on the screen icon in the lower right hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would like to now introduce today's guest speaker, Dr. Scott Kelley. Dr. Kelley received training in microbial diversity and megatronics from one of the founders of this research field, Dr. Norman Pace, as a postdoctoral fellow of the University of Colorado. He was also trained in bioinformatics by one of the earliest and most influential bioinformaticians, Dr. Gary Stormo. Since that time, he has published 53 papers related to microbial, metagnomics, and bioinformatics in many influential journals. His studies include both DNA sequence-based analysis of microbial diversity of numerous different environments, including the built environment, human gut microbiome habitations, numerous papers on bioinformatics software tools, and novel analytical approaches to data analysis. Dr. Kelley is a pioneer in the application of molecular NGS analysis of microbial diversity in the built environment. Humans in industrialized society spend greater than 90% of their time indoors, and we know precious little about the indoor microbial communities. Along with Dr. Norman Pace, he published the first paper that used cultural independent methods to study an indoor environment, namely shower curtain biofilms. Since then, he has studied a broad range of indoor environments, including microbiome, contaminated hospital pool air, and surface and neonatal intensive care units, offices, airplanes, daycare facilities, and restrooms. These papers are well cited and help inspire the Alfred P. Sloan Foundation's Microbes in the Built Environment program. I would now like to turn it over to Dr. Kelley for his presentation.
Good morning, everybody. Good morning, everybody from San Diego. Good morning, everybody. Scott Kelley from talking to you from San Diego, where it still is morning. So I'm going to be talking about, as Robert said, the serious science of Schmutz and deep sequencing in the built environment. And please forgive me if it's a little bit awkward at times. It's the first time I've done an interactive presentation like this, and I'll do my very best. Hopefully, I won't mess it up too badly. So I've been working on, as Robert said, I've been working on the microbiology of the built environment and metagenomics since 2001. And with Dr. Norman Pace, we published the first study of culture-independent study of the built environment, which means we're using sequence analysis rather than culturing on a plate. And I'll talk to you why that might be important later on. But essentially, we were the first study to really explore built environment diversity, and since then I published a lot of other papers, and the field has grown tremendously since that time. So let's get started. All right, so I really like this quote from a paper by Beazle et al., and this is also with Dr. Norman Pace, that humans, we move through a sea of microbial life that is seldom perceived except in the context of bacterial, of potential disease and decay. So it's all around us, it's all in us. We have 100 trillion microbes in and on our bodies, 10 times more than our actual cells. We know nothing about them, practically nothing about them, except the ones that actually cause disease and decay that we actually know about. So there's a lot more than we need to discover, and even if we could culture all of them, which we can't, it would take forever to actually get through them all. So really, by applying molecular methods, we've greatly expanded our understanding beyond the culture work. And this also includes the built environment, which, as you'll see, is increasingly important in human health and just in human existence. So the reasons to study indoor or built environment microbiology here's a few reasons. First of all, we spend westernized or industrialized countries spend just about an estimated 90% of our time indoors. And that includes increasing amount of the, what used to be the third world is now the industrialized world. I was recently in China, and I was in the city of Shenzhen, and in the last 30 years, it's gone from a population of 300,000 to 17 million people. So more and more people all around the world are moving into cities and spending all their time indoors. And I think something like children in the United States spend on the order of 11 hours a day in front of their screens. So that's the average. And that's an incredible amount of time to spend indoors. So you could really say that the indoor built environment is now the new human habitat. Disease transmission is also an important aspect. There's a large debate on the importance of inert surfaces in the spread of microbes. We know that you can pick up a lot of microbes when you can go to the hospital, including antibiotic-resistant ones, but the question is how long, where are they, how long do they interact in surfaces, can we move them from place to place, and how many opportunistic infections are we going to have on these. So it's an open debate, but I believe, and I'll show you, that I think that culture-independent methods can really, or deep sequencing methods, can really shed some light on this, and in combination with other studies can really help us understand how things are moving around and what that means for health. Again, opportunistic infections, there's something on the order of 100,000 deaths per year in the United States alone that come from being in the hospital. So that is an enormous and terrible statistic. And this is inside a built environment. So what do the surfaces, what do the buildings have to do with this? Clearly, there's also sick building syndrome. It's been talked about for a long time. Some people get sicker in certain buildings than others. What is the role of the building itself or the conditions of the building or the people inside that lead it to have a lot more microbes, particularly fungi, that can grow in these buildings. And there's a lot of storm damage and other things, so we need to understand that. And frankly, it's just a fascinating environment in its own right. When you talk to building engineers, when you talk to people who really understand buildings, they're fascinating and diverse environments, and they are worthy of study in their own right. Molecular methods. So the molecular methods have really opened our eyes to the microbial world. It's been an explosion of information and understanding, certainly about the human body, but also all environments, including the built environment. Most of the microbes are just unknown. Much more than 99%, 99.999% have yet to be cultured of any sort. So we do not know how to grow them on the plate, which doesn't mean they will never be grown on a plate. That's how we can learn a great deal about them, but they haven't been yet. And so in order to study anything about them, we really need to get their genetic sequence. Unclassified organisms, there's outrageous number of classified organisms that have been identified only through a particular marker gene, namely the 16S ribosomal RNA sequence. Now that we're expanding it, we're also using other parts of the sequence and stitching whole genomes together. But it's still true that there's many groups of, entire groups and clades of microbes that are only known through the 16S, through a single gene marker that we can get through some of our molecular techniques. So there's a lot more to be understood. And what we can do is we can use the unique DNA sequence patterns in this gene and other genes, but in this gene and other marker genes, to ask several very important questions. Who is there? Who are they related to? Why are they there? And even where did they come from? And I'll show you some of the ways in which we're able to do that. All right, so the solution to not being able to grow them all, or it's a solution, but to still be able to study them, understand who they're closely related to, and maybe even their function, is the fact that they all have genetic material. This is genetic material if you're talking about bacteria and a lot of viruses, also things like archaea and fungi. The genetic material is DNA. DNA is really easy to work with. It's easy, and all DNA behaves the same. All the organisms may all be different and grow in different conditions, but once you break open their cells and extract their DNA, it all dissolves nicely in water or other solutions, and then you can study it very easily chemically, and it's extremely stable. So what we're going to do is take the entire, what we tend to do is we take an entire microbial community or assemblage, and we break open all the cells. That's the first stage. First, we use chemically or mechanically shear open the cells and the cell walls, releasing all the contents of the cells. Once those are released, we can purify out the DNA, get rid of the proteins and the lipids, leaving only this DNA or RNA. And then once we have the DNA, especially the DNA, you can do this with RNA too, but you have to turn it into DNA. Then you can take out the DNA sequence and get all the chemical letters using deep sequencing approaches or otherwise. These days, deep sequencing. You're going to see in my earlier studies, we cloned it and sequenced it, which was very tedious. But now we can get the letters of the sequences, which work wonderfully in computers, because then you can use the patterns of letters to determine who's there. So instead of storing all this information, we can just store the letters of one strand of the DNA sequence and put it in the computer, which is great at storing these kind of information. And then we can use these as a sort of barcode, if you want to put it that way, to type the species and figure out who they're related to. So for instance, down here in the bottom left, let's see if I can use one of these devices. Down in the bottom left here, we see a FASTA file, and here's a particular sequence that we've grabbed from the extraction of DNA in this environment. We sequenced it. It came out in our computer, and we can use techniques such as BLAST or other comparative analysis programs to ask which bug did it come from. So for instance, this sequence could be this particular organism in that environment. This sequence could be of this purpley spiky organism. Here's another organism that has this sequence, and the fourth one is the sequence over here. And we can actually count the number of sequences that come from that are of this type, and we can group them together and ask how many or relative proportions of this organism in this particular environment, and do that for all the organisms that we can get the sequence for. So it's a really effective way. I mean, even just having these 4 organisms would take a long time to culture. If there's 1,000 organisms there, you can just forget about the culturing. So the sequence information becomes the primary way for us to understand who's there. Okay, so the methods, I'll just go over quickly the methods I'm going to be using in this study. We're going to, all the methods are based on direct DNA extraction of microbial communities. The RNA comes out with, I'm not using any RNA techniques, but RNA can be used to study things like transcription in the environment, and as well as RNA viruses. I'm going to show you some studies that are based on single marker gene sequence where we use conserved primers to amplify all the same marker gene from all of the bacteria, say, in a particular community. So it'll amplify the 16S ribosomal RNA gene sequence, and because it's a conserved primers to a site that they all have, we can PCR amplify it from all the bugs in the environment, and then we can send it off for sequencing and then analyze it. I'm going to be using I'll also show you some epifluorescence microscopy, where a DNA and RNA stain that actually intercalates into cells and viral-like viruses or viral-like particles, and allows you to visualize without culture and count how many bacteria and what we call viral-like particles there are on a slide. And then I also have data on shotgun metagenomics, which is instead of amplifying a single sequence from all of the different bacteria, you take all the DNA, cut it up into little bits, and sequence all the fragments. And then you can either analyze the little fragments of DNA separately, or you can stitch them together to try to get large contigs or even whole genomes. But it's a way to not only look at the taxonomic profiles of who's there, but also the functional profiles. And when we're done with all this, of course, then we send it off to the nerds and give them some pizza and a few Skittles, send it down into the basement, and get some analysis back about what's going on. Right. So first part, I'm going to be reviewing some of my built environment studies. Just go through a history of it, and it'll help you understand, hopefully, a little bit of the why, how we started it, what it means, and what it's useful for. And I'll select a few interesting stories based on both on 16S loan libraries. Whoops, missed a C there. Both on 16S clone libraries and some deep sequencing studies as it progressed. Back in 2004, we had clone libraries, and it was just getting started with next generation sequencing. So that's what we used. But it turned out to be extremely effective, just a little tedious. All right, so I wanted to talk a little bit about one of my favorite early studies that that was really says a lot about the built environment. But this is about a hospital therapy pool that was where the lifeguards of the pool were actually getting sick and coughing up blood. That sounds really terrible. So we suspected that they were perfectly healthy lifeguards. The people in the pools weren't healthy. That's why they were in the pool in the first place. But the pool is entirely contained indoors, and it's warm at a nice, roughly body temperature, and it's got sick people swimming around it. And they couldn't chlorinate it like they wanted to, because the chlorine itself, if you sit there as a lifeguard, would actually damage the lungs. So we wanted to know the lungs of the lifeguard. So sort of a situation of damned if you do, damned if you don't, really. Anyway, so what we did, I worked with an engineer, and this is also in the PACE lab. We sampled sterilely sampled both the air and the water of the therapy pool. We extracted DNA directly from the water or the air samples. Wasn't that easy. And then we PCR amplified with our universal primers for the 16S gene, and we sequenced it. Sequenced the clone libraries. Now, what do we find? So this is a complicated slide. I apologize for this, but these are each pie charts. Here's an example pie chart that comes from it shows the phylogenetic diversity of the organisms from a single sample. In this case, this is a water sample DNA extraction. In fact, B, C, D, E, and F were all water samples or samples on the side of the pool. And you can see that there's quite a bit of different phylogenetic groups. Each one of these little pies indicates the proportion that was gamma proteobacteria groups, or other bacteria, or mycobacteria, or sphingomonas bacteria, and beta proteobacteria, and so forth. And what I wanted to point out to you is the water samples were really quite diverse and highly variable. But then we did the air sample. In the pool air, we had a completely other type of response, that it was entirely dominated by mycobacteria. And mycobacteria, well, there is mycobacteria tuberculosis. Now, this wasn't tuberculosis. We didn't find any. But it turns out that other mycobacteria live in the soil. They probably like to grow very nicely at 37 degrees, and they're being enriched in the air, which is probably leading them to get into the lungs of the lifeguard. So they're not normally pathogenic, but at high enough concentrations, some of them can have similar effects and actually cause lung problems if in high enough concentrations. And we found a whole lot of mycobacteria, like 50 different species that were enriched in the air in this one study. Almost none of them had been cultured. So they were all known by sequences at that point. So very interesting application of the method. Another application of the method we used was in the NICU, newborn or neonatal intensive care units. I did one of the first studies of this using culture-independent methods. Culturing methods have been done for a long time, but we wanted to know how easy was it to find and what types of microbes would we find on all of the surfaces that were frequently touched by the staff. And you can see in this NICU, which was very much like the one we studied, there's loads of people in there touching all the equipment and going towards little babies. And this is an important area to study because the little babies, their immune systems aren't well developed. They have a lot of tubes in their bodies and they're very sickly. So even if it's not a pathogen, opportunistic infections are extremely common and have devastating consequences in environments. So we really wanted to see what was there and how many different types of bugs were found in this. And so when we did the analysis, here's a breakdown of the analysis. Just quickly, this is a principal coordinates analysis of where each dot here is a mathematical representation of a particular sample that we sequence. So this dot up here, the blue one, let me use this for instance. Getting used to the technology. Okay, so for instance, that is a representation of the entire bacterial community that came from a single swipe sample on, say, one of the computer keyboards that we were studying from NICU, first NICU. And all the red dots come from the second NICU. To interpret these plots, you really have to say if the dots are closer together, the community as a whole, those two samples, their entire communities are more similar to each other. So for instance, if you're looking at these two samples, they're almost occupying exactly the same space. Their communities are highly similar. So first of all we found when we did analysis is we found all sorts of sequences that were closely related to known opportunistic pathogens. We found evidence that there was Neisseria, Pseudomonas, Streptococcus, Staphylococcus, and lots of different bacteria. We don't know if they were living or dead, but they were. A lot of them can survive for long periods of time on dry surfaces. But we also found an interesting pattern that most of the NICU of the two environments broke down together, but at one part in one of the NICUs, the blue one, we found a lot of samples that were completely dominated by Enterobacteriaceae, which are often known to be gut bacteria. So at some point in time, we feel that there was some kind of spread of these Enterobacteriaceae, and we know there is a lot of diaper changing and other things going on, of course, in this office. But all of the surfaces were suddenly dominated by this group. And they really were different from the other NICU and some of the other, the samples from the other NICU, but also the samples from the same NICU. Then we did another analysis where we took all those samples and compared it to all of the known office studies or built environment studies that we had at that time. This is another principal coordinates analysis in three-dimensional space. And what you can see, and I know this is very complicated, but I'll try to break it down easy, is here is right here is this area. Most of the studies, it doesn't matter if they're offices, NICUs, and restroom surfaces, all fall out into this area here. But all of our NICU studies from that one study are far away from all of the other office and studies, which means this was a particularly interesting event that happened only to this NICU. So this is a way that we can tell that maybe deep sequencing approaches could help us understand some event or infectious spread that might have happened inside an indoor environment. All right. Airplanes. So I also did a study. In fact, I should point out that this in this particular study in NICUs has been followed up very nicely by some other people who actually showed that organisms within the newborn infant intensive care units can actually colonize newborn infants that are born inside the hospitals through C-section. The C-section babies can actually be colonized by microbes that are in the NICU. All right, so airplanes. I also did a study on airplanes where I was able to sample several different domestic and international flights, actually 3 domestic and 1 international flight, and look at what happens to the communities as you go through. So essentially, before people show up, the whole airplane is really nice and clean. But then once these people show up, it gets really filled with microbes very quickly. I did in this study, I did 4 different sites. I did many swabs on armrests, tray tables, and a lot of them in the laboratory. So and each flight I did a repeat of all this. 1 international, 2 domestic flights. I swabbed. I brought them back at room temperature, and I definitely avoided alerting the TSA. At that time, it wasn't a big deal. And then I did the clone library and the sequencing work. And what I found was almost all the action. Oh, sorry about that. All the action was taking place in the restroom. Every surface in the restroom was contaminated primarily by things like propionibacterium, staphylococcus, and streptococcus. And interestingly, corynebacterium was pretty prevalent. And that is associated with prostate infections, although I can't say that they were causing prostate infections. They may have come from that. But yeah, the laboratories were just covered. Even the paper towels had significant evidence of microbial diversity. All right. So now on to a second part of my talk. Now that we know, and we've known this in a lot of studies now, it's repeated consistently, we would really like to know, we know that microbes are on every surface. A lot of them are brought in by the humans themselves, although there's also evidence of environmental bacteria. And one of the main questions I wanted to know was how fast do these assemblages actually form? How fast do you get these microbes colonizing? And even, is there any kind of ecological succession where you start off with a certain kind of microbes and then you end up with another altogether? So in this study that I'm going to concentrate on now, which is pretty recent, I'm asked 3 questions. How quickly do assemblages form? How stable are they long term? Do they go to the same place every time? Are they always random and different? And how consistent are they in their composition? So how did I go about doing this? Well, I did this by, I focused on restroom biodiversity. So this is all on the restrooms. I would say, I think it's going to be similar to other environments, but I'm going to say this is a particular restroom here at the university. And I did a time series analysis of the restroom biodiversity. And I call it that because I think biodiversity is usually termed as an ecological setting where organisms interact with each other. It's not clear that that's the case in indoor environments because they might just be settling there and not living together in the community unless they're a true biofilm. But sometimes I think they might be, but sometimes not. Anyway, that aside, we looked at 4 different restrooms at San Diego State University, 2 female and 2 male. And then we had 2 high and 2 low use. So the high was about 10 times as many people using as the low. We sampled the toilet seat, the floor in front of the toilet, and the soap dispenser pump. And I focused on the floor in front of the toilet because we ended up doing a lot of our work there. We did 1 time series with all 3, and then we focused on the floor for the rest of the study for other reasons. And we repeated it. We repeated it once a week for 8 weeks, the first setting. The first thing we wanted to do, though, is to actually find out, to actually sterilize and completely free it from all of the microbes. So we used epifluorescent microscopy to actually ask how many microbes did we see on these surfaces, and whether we could get rid of them to start completely from scratch. So what we wanted to do was say there was no trace of microbes, even DNA or RNA, dead or alive, on any of these surfaces, and then we would start running the time series. So first thing we did was we sampled each of them and did microscopy, and you can see that every surface is loaded with little pricks of light. The medium-sized ones are bacteria. The very tiny pinpricks of light are viral-like particles, and then the giant ones are cells. They could be human or fungi. Right. So what we did was here, first thing was we sterilized them using 10% bleach, and we actually had to, we were testing it at 0 minutes, 2 minutes, and 20 minutes. And by 10 to 20 minutes, we had no evidence of any. We went from lots of stuff to nothing. It took about at least 10 minutes, and I think up to 20 minutes of just leaving it sit there to completely eradicate all of the microbes and viral-like particles. So once we did that, we started running the experiment. All right, and so we actually ran a repeated experiment of how long it took to form these communities. So it's not a true time series, but what we would do is we would sterilize it, let it go for 8 hours, and then resample it. So we had nothing, 8 hours, sampling. Despite only 8 hours, we had perfect correspondence to a study that was done by Flores et al. In 2011 on restroom surfaces. So what they found in 2011 was that the soap dispensers, there was clearly distinct communities between the soap dispensers, the toilet seats, and the floors. And you can see this in this PCOA plot, principal coordinates analysis. All the toilet seat samples came together. All the sink samples, the hand soap dispenser samples were a unit, and all of the floor samples were also always together. And this took 8 hours to find the same thing that they had, which had developed over months and maybe years. They're clearly distinct communities and surfaces, and what was interesting for the relator is that the floor had the highest diversity of samples. So we concentrated, then we did a follow-up study looking over an hourly time point, a very short period of time to see how, so 8 hours we got a community that might as well have been 8 months. So it must have developed much faster than that. So we went back, we sterilized it again, and we watched it over the 8 hours, sampling every hour. And we only did the floor because of the time. So what happened when we sampled every hour from 8, 9, 10, 11, 12, etc.? What happened to it? Well, this is what we have. We actually used a SourceTracker program that I developed with a few other people to ask where the communities came from. So at the 8 o'clock, we sterilized at 7. We let it run to 8. We decontaminated thoroughly. It's not just sterilization. We let it run to 8 o'clock. And this is the proportion of organisms from the different environments that we found. At 8 o'clock, the vast majority were located to fecal bacteria. This is the floor in front of the toilet seat, so this makes a lot of sense. Then, so even though it was on the floor, skin bacteria were playing a fairly prominent role. So about 20% were skin bacteria. And then unknown, outdoor and unknown were the rest of it, 10 to 15%. That stayed constant pretty much throughout the time of the experiment. But what you notice is it goes from 8 to 9, the fecal bacteria are getting smaller and smaller and smaller proportion all the way down till about 3 o'clock, it looks as if it had been 8 weeks. So by about 3 o'clock, it's fully, the diversity is about, after a few hours, is as high. And then the succession pattern happens over the day, and by 3 p.m., it might as well have been 8 weeks. So it was a really pretty rapid assemblage of organisms. And it happened faster, of course, in the high throughput, high diversity restroom than the high occupancy restroom than the low occupancy restroom. All right, looking at it a different way, you can see that this is another way to the successional trajectory over time in the principal coordinates plot from, I have it going the wrong way here. All right, this is the successional trajectory and the principal coordinates plots, looking at the frequency and as it moves from 0 hours to 8 hours, and then over time it reaches a peak that is slightly different, but very similar to the final 8-hour time point. So to get to the final diversity, it takes longer, but most of it's already there. All right, this is also an interesting part of the study. We found a clear separation of these are another principal coordinates analysis, from the female and the male restrooms. So there's quite a bit, a clear distinction between the bacterial diversity in female and male restrooms. The split in the points is as clear as it usually gets in these type of plots. And when you look at the individual bacteria, the relative abundance of individual bacteria from these two samples, you can see that there's the female restroom they had a big dominance. Well, it's not a huge proportion, but the things that were really different between them were lactobacillus, which makes a lot of sense, and it switched with the men. Rosaburia was higher than with the women. I also noticed that the Blaudia was also different, but it was really a few species that were really driving this change. Would also make a lot of sense. The high and low usage was also different, not quite as distinct. Here's the high usage in red and the low usage in blue down at the plot. There's pretty good separation on the PCOA plots. And what we found was that if I look at the, I'll point it out here, the bacteroides was higher in the high usage rather than the low usage plots. Bacteroides is associated with fecal samples, and Arapisca, I can't even say this one. Erysipetracea is also known as a gut organism, and it tended to be much higher in the high usage than in the low usage things, probably because the anaerobic bacteroides probably dies out pretty quickly, leaving only the skin bacteria left because it's anaerobe and it doesn't survive in oxygen. But if you're constantly having people come in and use the facilities, then they're going to be refreshing it with fecal bacteria quite consistently, whereas in the low usage they tend to die out more and skin bacteria are dominating. We also saw that the Sphingomonas were different and higher in the high usage, and those tend to be water-loving organisms. So when you're doing a lot of usage of the restroom, water from either the toilet, I know it's disgusting, but that's the way it is, or from the sinks is probably being sprayed around a lot more in that particular environment. All right, I also have some data on viral abundances and diversity. This is on the top here, I have, sorry about the figure legend, but it's the opposite. On the top here, the Shannon diversity we saw went up from 8, 9, 10, 11, 12, so the richness of the community is steadily increasing in terms of the bacteria over time. And then we also looked at the viral abundances versus the bacterial abundances. And we found that there was a tight correlation between the amount of bacteria in the surfaces and the viruses, viral-like particles when we were doing the counting. Interestingly enough, though, in many environments, the viruses are 10 times as numerous as the bacteria. So in oceans or in living systems, the viruses are actually more abundant than bacteria. But in our situation, the ratio of viruses to bacteria was 0.4 to 1, which is a really low ratio. And it does indicate that the dry surfaces are not very actively the viruses aren't very active on these really dry surfaces. When we looked at the shock and metagenomics, one of the things that jumped out at us were the viruses, especially the DNA viruses associated with not only bacteria, the bacteriophage, but also the human viruses. And what we found were two groups of organisms, including the herpes viruses. Herpes all over the restroom, right? Actually, I just found this when I was in Germany recently. They had a whole public campaign about going to the doctor if you had some infections, herpes infections, and so forth. So, you don't see these kind of advertisements in America very often. I thought it was quite amusing. Anyway, the herpes viruses were all over the restroom, and I'm not exactly sure what they were. There's a bunch of different families of herpes viruses. But we also found a lot of HPV, herpes, or human papillomaviruses all over the restroom. Most of them were skin and wart causing, either plantar warts or skin wart herpes, including a lot of them had not been cultured or identified. And that's really interesting personally because I had a kidney transplant a few years back, and it turns out that people who've had kidney transplants are very susceptible to getting warts, inflammatory warts, probably because their immune systems are so suppressed. So understanding that the viruses and herpes viruses or HPV viruses are everywhere makes me think again about wearing flip-flops every time I go in the restroom. Right, so the last part of the talk I wanted to go over was the part about microbial, I titled Microbial Dispersal. But it's really about some of the software methods that we've used to figure out essentially where the microbes came from that are contaminating built environments or practically any environment. And so we invented this method that's based on Bayesian statistics called, it's a Bayesian community-wide culture-independent microbial source tracking. And the other title is, Where Did That Schmutz Come From? And this is what it does. So the inspiration for it was the indoor environments. DNA surfaces of when you look at the bacteria that are on surfaces, say for instance in this daycare study that I did, it was a mixed bag of bacteria from different sources. You could just look at the species and you knew that some were from soil, like bacillus, some were fecal bacteria, other were skin, propionibacteria for instance, and then there were other ones we didn't know where they were from. So obviously they're from not a single environmental source. They certainly don't grow naturally on children's tables or on these surfaces, but they're there nonetheless, and they had to come from somewhere. So the question was, what are the sources, and how much does each source contribute to the entire community? So the question is, where does that schmutz come from? First, it's not native to a sample. It didn't grow there. Second, it's mixed, likely multiple potential sources. So we came up with a posteriori test that compared the sample data set we had to other sample data sets from other sources. So for instance, we have an indoor environment, and here's a skin one we're comparing to, and here's a fecal one, and here's a soil one, and here's something else. And we even had an unknown one that we could say, we don't know what these are. But we can use it to determine the source of the contamination and also partially track environmental contamination or the movement of microbes. So the Bayesian approach takes a Bayesian model of uncertainty about known and unknown source environments and use a Gibbs sampling procedure to sample to create the distributions of where it came from. So your sample X is considered a set of n taxa, which each one can be assigned to a source environment of some sort. So you have to have the source environments. And this is the paper if you want to go into more detail on that. But here's an easier sample. So let's say we have an office desk. We're going to call that our sink community. And our sink community is composed of bacteria from all sorts of sources, including my hands when I touch the surface here. And then we compare the sequences that we got from that sink community to a bunch of other samples from other studies, or even our own if we want to sequence it, that could be contributing to that environment. So for instance, we get a skin sample, a mouth sample, a gut sample, a soil sample, and then we leave a spot for, well, we don't know where it's from. And then we ask what proportion came from the skin, what proportion came from the mouth, gut, soils, etc. We could also do this with other things. If we have a sink community of a contaminated water source or anything else, we can say, well, which did it come from? Did it come from cow poop or bird poop or human or sheep? Is it human sewage that contaminated? And that's also a useful way or another use of this software. Yeah, in the sense that it's contaminated food, we'd really like to know, did it come from some farm where they had cows or what? All right, so here's an outline from the paper that showed us how we can get these data for a particular environment. And this was used to ask we used this in the restroom study. And I'll show you an example. This is really fun. This is our contaminated lab PCR water that was making amplifications of bacterial sequences when it shouldn't have. And we found that, in fact, most of the higher proportion of it came from gut sequences. So somehow somebody had contaminated from gut microbiome bacteria or fecal bacteria somehow got in the PCR water that was actually sent with a kit. I'm not going to tell the manufacturer. And then some of it was skin contamination and a little bit of it was unknown. Here's the NICU, the newborn intensive care unit. Most of this was actually contaminated from, I'm going to use the little markers here. The red indicates that most of that came from skin. So that makes a lot of sense because people are constantly touching this environment, although there was a fair amount of unknown in the sample as well. And then the office phone, again, a lot of it is going to be skin bacteria, and we found that about 50% of this particular sample was skin, where another, interestingly enough, was from about 20% came from soil bacteria, with the rest from an unknown source. All right, so it's a very useful technique for understanding how things develop and where they come from in the built environment, or as it turns out, many other environments. It can also be used the method can also be used to determine species frequency, so how many of which kind of bacteria are part of that environment. And I'll give you an example of this. So in the NICU, or let's do the office study actually. In the office study, we can tell you that a lot of it was skin bacteria, and that skin bacteria primarily, we said it was skin bacteria because it's propionibacteria. That makes perfect sense. We also we get another instance of the red here was, we can actually not only tell you where it comes from, but what bacteria is represented in that particular environment. In this case, Pseudomonas is a big player. But in the NICU, for instance, enterobacteria from the skin was a big player that said it was, in fact, it was located for skin for some reason. Right. And since then, it's been used a lot. I've been really happy to see it's been used in a lot of publications that go way beyond the built environment. So, for instance, it was used in a microbial signature to look at fecal pollution in Michigan. It was used to ask if cohabitating family members share microbiota with one another or their dogs. It was used to look at, in a longitudinal analysis published by Jack Gilbert and colleagues, about looking at humans in the indoor environment, so how they contributed it over time. It's been used in intensive care units because it turns out that newborn intensive care unit, the microbes from the guts of newborn neonates in the intensive care unit actually resemble those in the unit, resemble those found in the infants themselves. And it actually was also used to show that in a mouse study that bacteria, which direction the microbes were moving from one mouse to another, because we knew one was the source and one was the sink. All right, and then in the next stage, we're working on making it not only work with the 16S data, but all sorts of other data, which I'm really excited about. For instance, we're going to use it. We're going to use, right now it works with 16S data, but we're going to adapt it for shotgun metagenomic data as well. We're hoping to do it, expand the method so it can also use with chemical methods like mass spec. And we're trying to make it work not only on bacteria, but also viruses and fungi. So SourceTracker can be used with all of these things to track potential sources of contamination. All right, and the idea is that we're going to get some greater specificity, more unique signatures, and more biological insights by using many more sources of information than a single marker gene study. All right, and for acknowledgments, here's my collaborators. There's so many I have to thank. I put a set on, but I do want to especially thank Tara Schwartz for doing all of the work on the restroom study, because that was a big part of this, and the sampling, UCSD Hospital and SCSU, and the funding from the Clorox Corporation, and especially the Alpha P. Sloan Foundation and their Built Environment Program have been instrumental in moving this forward and my research forward. And with that, I'll take any questions. Thank you.
All right, and thank you very much for your presentation. Hey, before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we'll try to answer as many questions as we can. So Doctor, I do have one question here that's queued up for you. Can microbial environments detected in traditionally chlorinated pools grow in saltwater-based treated pools?
So do not. So sorry, I don't know if they've tested that. Actually, that's a good question. I imagine that they would not grow as well because of osmotic pressure, but we have not tested the biofilms on saltwater pools. My guess is that the saltwater would be very effective at keeping down the bacteria. But I have not done that study, and that's actually pretty interesting because we have some in the area. That'd be really, really nice to be able to study that to see if that was an effective way of cutting down both the amount of chlorine that's been used and the bacterial loads themselves. So that's an excellent question.
All right, and thank you. And this is going to be in response to your hospital pool water and air bacterial study. Have you done a comparison versus a standard community pool environment? Curious on how that might have a differ especially in the microbacteria concentration.
I have not done the. I have not. Oops. I've not done the study with in comparison. I'd love to do it. Need some money, unfortunately. If it's not, my guess is that the community pools tend to be a much colder, cooler temperature, and they tend to be chlorinated. So when they chlorinated the pool after we worked on the study, the bacteria levels went down tremendously. Also, if they were salt water. So the temperature difference, I think the microbacterium love to grow at this higher temperature, but also open pools would definitely be different. The closed environment, the high humidity, and the lack of and the chlorine would probably make a big difference. But to be honest, I haven't done that study. All the ones we have here in San Diego seem to be outdoor pools. Well, there's a few indoor community pools, and that'd be really interesting to do that experiment. I think I'm going to propose that next. Thanks for the idea.
Alrighty, here, and this is our last question that we have time for today. And doctor, is 8S or 14S sequencing better for bacterial identification?
You know what 8S? I think there's 16S and 18S. So 16S ribosomal RNA and is how they refer to the small subunit ribosomal RNA gene sequences that are in bacteria. The small and the 16S is a size marker of how it runs on a particular gel that's from a long time ago. The 18S is also the small subunit ribosomal RNA sequences, but they are larger and they're from eukaryotes like fungi and humans and mice and all that, and trees. So they're just a larger molecule. And it's that particular marker gene, the ribosomal RNA, is really great at telling apart bacteria, archaea, and the 18S, the same marker, can also be used for things like fungi, but it's not, as for the fungi, it's not discriminatory at a very low level. So the sequences are very similar to one another among the fungi, and you end up have to use another marker called the internal transcribed spacer, which is between the, I think it's between, well, it's anyway, I think it's between the 16S and 18S markers to be able to identify them. So it's really good for bacterial identification, but it's okay for fungi. It just doesn't tell you it doesn't tell you species or even genus. I think it's more like at the family level.
All right, great. Thank you so very much again. And do you have any final closing remarks that you would like to have?
Yes. Thank you. No, I just like to appreciate the people for setting up this LabRoots seminar and the organizers of the committee, and thank you for everyone who's attending and for your really interesting questions. I appreciate it.
All right, thank you. And with that, I would also like to thank the doctor for his time and his presentation, along with LabRoots for allowing this presentation as well. Today's webcast will be available for on-demand viewing through December the 8th. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's event. Until next time, goodbye.